Well, greetings. Welcome, everyone, to PCAP's Native Prairie Speaker Series. My name is Caitlin Rose Seiler, and I am the Stewardship Coordinator with Saskatchewan Prairie Conservation Action Plan, or SKPCAP. Today, we are really lucky to have three expert speakers talk about scale-dependent relationships between habitat, insects, and birds in native grasslands. Before we begin, I'd like to state that we respectfully acknowledge that we are on traditional territories of many Indigenous nations and communities, past and present. For millennia, they've worked to protect these landscapes and the life these areas sustain. I'd like to thank these original caretakers and acknowledge the ongoing work and presence of Indigenous peoples in Canada today. And just a few items before we get going, I'd like to note that PCAP's Native Prairie Speaker Series is a monthly presentation about anything to do with native prairie conservation or species at risk. On May 23rd, Annabelle Dombrow, a recent graduate from the University of Alberta, will be speaking about the management of annual brome invasion within northern mixed grassland using indaziflam. And on July 30th, Mike Blom will be speaking about migratory birds at Redberry Lake Biosphere Reserve. So stay tuned for details about our June webinar, which will be announced shortly. And a reminder to all of our listeners out there, you'll be muted for the duration of the webinar, but if you have any questions during the presentation, just type it into the Q&A section of the webinar dashboard at any time during the presentation, and feel free to upvote each other's questions if you have the same one. I'd like to take a moment to note that financial support for today's webinar is provided by our presenting sponsors, North American Helium, Saskatchewan Cattlemen's Association, Nutrien, SODCAP. In-kind support is provided by Birds Canada, Canadian Wildlife Federation, as well as SODCAP. And I'm pleased to introduce today's presenters. First of all, Diego Steinmaker will be speaking first, and he is a biologist with the South of the Divide Conservation Action Program, and with 25 years of experience in ecology and management of temperate grasslands in South and North America. At SODCAP, Diego works with ranchers and other organizations on the development and implementation of rangeland conservation and restoration programs in southwestern Saskatchewan. Dr. John Wilmshurst will be speaking second, and he is the Native Grassland Conservation Manager with the Canadian Wildlife Federation. He is an adjunct professor in geography and planning at the University of Saskatchewan with 30 years of research, monitoring, and communications on grassland ecology and conservation. Among other projects, he coordinates the CWF's activities in the SSGF and the Weston Family Foundation Multi-taxa Project and the SODCAP Living Lab Central Prairies Project. And our third speaker today is Dr. Kevin Hawkshaw, the Prairie Program Biostatistics Analyst with Birds Canada, using data to support the conservation of grassland birds and their habitats on the working landscapes of the Canadian prairies. And central to his work is the development of the Bird Friendliness Index, a bioindicator used to measure the value of working landscapes for grassland birds. He also supports the SSGF Multi-taxa Project and the SODCAP Living Lab Central Prairies Project. So today, Diego will be speaking first. And unfortunately, despite all of our test runs, we haven't been able to get Diego's webcam working, but Diego, you can go ahead and share your screen and take it over from here.
Yeah, good. Thank you. Yeah, I'm sorry, my camera is not working well today, but the most important thing that you will be able to see the slides and the results. So I will start presenting our research questions and goals and how. Yeah, good. Thank you. Yeah, I'm sorry, my camera is not working well today, but the most important thing that you will be able to see the slides and the results. So I will start presenting our research questions and goals and how we evaluate vegetations and habitat variables. Then John will continue presenting the methodology for insect collections and results that relate with vegetation and insects. Kevin will integrate all the taxa, that is plants, insects, and birds. And finally, we will finish with some conclusions and take-home message. This project born from an initiative of the Stock Growers Foundation that had funding from Weston Family to investigate the relationships amongst different taxas in native grasslands. Today, we are just us present in this, but many people participate in this project. We will list them at the end of this presentation. I want to introduce briefly SODCAP to those who don't know us. The South of the Bioconservation Action Program is an NGO that, in partnership with other NGOs, governments, First Nations, ranchers, and industry, has been delivering programs during the last 10 years to restore and conserve native prairies in southwestern Saskatchewan. SODCAP is also leading the Living Lab Central Prairie project, along with many other partner institutions. With this mandate, SODCAP is participating in this project to investigate how vegetation and habitat impact on biodiversity. We know that grazing and other factors such as climate and fire affect vegetation and habitat for wildlife. And particularly, we know quite well the relationship between habitats and birds. For example, we know that species like spruce pipit or bear sparrow prefer grassland with intermediate vegetation height and good amount of litter, but other species such as thick-billed longspur and chestnut-colored longspur prefer shorter or sparse vegetation. Then we can manage the habitat with grazing and fire if we want to favor some species. However, we don't know too much about the role of insects in these relationships. That is, how plants affect insects and birds in this grass. We assume some type of dependence among them, since insects use the habitat and are food for many birds. But again, we don't know exactly how they interact among each other. So in this project, we set plots, circular plots of 400 meters in diameter, 31 acres in total, where we evaluate vegetation, insects, and birds all in the same site and the same moment. In every case, we select the site taking into consideration some restrictions, such as the distance to the water source, that must be between 600 and 800 meters, distance to fence and roads that have to be more than 400 meters, excluding sites with extreme slopes. These assessment plots were distributed in different ranches across the prairies in southwest Saskatchewan, in the Middle River watershed. We would like to acknowledge the land on which we work is Treaty 4 territory, so we pay all respect to the First Nation of this place. And this area represents an important reservoir of grassland biodiversity because provide habitat for many species, including more than 20 at-risk species that depend on these grasslands. In 2022, we select 32 plots in 5 different ranches. And in 2023, we evaluate 20 sites in another 4 ranches. And we have planned this season to set 26 plots in other 5 places. These places include, again, private ranches, community pastures, like Mancora, Dixon, Clayton, First Nation reserves, Nicanit First Nation Reserve, and Grassland National Park. These sites represent different ecological sites or ecosites, such as long, thin, sand, sandy loam, dunes, ecosites. Ecosites are defined by local factors such as topography, soil texture, moisture, salinity, that create different environments for plant growth. Here, today, we are presenting the results of the 22 and 23 assessment. We characterize the site by measuring different vegetation and habitat variables. We measure habitat traits related with plant species composition, some variables related that measure vegetation structure, such as, for example, grass height, standing dead grass, rubber pole, and variables, and also variables related with hydrological function and soil protection, such as, for example, litter amounts, cloud moss cover, and background. All these evaluations, the habitat and vegetation evaluation, were carried out by technicians from SODCAP and Stock Growers Foundation. Now I would like to briefly describe one of these variables, because in a few minutes, Kevin and John will show some results that relate this variable that is called visual obstruction readings with insects and birds. E of R is measured with rubber pole and is a good indicator of standing above-ground biomass, and a good measure also of vegetation structure because combine vegetation height and vertical density. At each sample point, up to 4 readings are taken from 4 meters from the pole, and readings are all bands which are obscured or hidden by vegetation. In addition to vegetation, the average of these vegetation variables, we also consider the standard deviation of the mean. A standard deviation is a measure of how dispersed the data is in relation to the mean. That is high or large standard deviation indicates that data are more spread out and is more variable. So we can use the standard deviation as a measure of heterogeneity. And we consider that this standard deviation because there are many studies that found some relationship between heterogeneity and biodiversity. So we also try to answer this question in this project, that is how heterogeneity of the habitat relate to biodiversity. In different situations, in different sites, we can have more homogeneous or more heterogeneous areas, more patchy sites, and then we ask how patchiness affects biodiversity. For example, in that specific case, we have this plot that have different patches with shore vegetation, with one snowberry clump, with intermediate grassland height. So we want to, we evaluate with a measure that is standard deviation, the degree of heterogeneity in the sites. For example, comparing these two sites, we have that in one site for one given variable, for example, rubber pole, the mean is the same, is 4.2 in both cases, but the standard deviation is totally different. So in one case it's more heterogeneous, or we have more vegetation structure variability than in the other or the other case. So in short, we evaluate different vegetation and habitat variables in different ecosites and with different range conditions. In each of these plots, we evaluate, again, species composition, vegetation structure, soil protection, in all the different areas, in some long dunes, in long ecosites, and thin ecosites. And we get different sites with different range conditions, with different vegetation structure, with different amount of litter, for example. And we ask here how these different conditions, different situations relate to insects and bird communities. So I will leave here now, and I will pass the ball to John and Kevin, who will show how these vegetation and habitat traits relate to insect and bird communities.
Okay, well, thank you very much, Caitlin, for inviting us for the webinar today, and thank you everybody for attending. It's a bit overwhelming, the big crowds that we get with these PCAP webinars, but we're really grateful for the opportunity to talk to you about our research. So I'll skip on to we've already sort of seen most of this introductory material. So my name is John Wilmsers. I'm with the Canadian Wildlife Federation and manage the Native Grassland Conservation Program. And the Canadian Wildlife Federation was taken on the responsibility of, in the Multi-taxa program, working on the insects in this whole great network of ecology in native grasslands. Why were we interested in insects? Well, I mean, insects are obviously a really essential part of the biodiversity of native grasslands. They are affected by the kind of management that we see often in native prairie. So they're affected by cattle grazing, by vegetation structure, and by the types of different, as Diego pointed out, the different types of grasslands that we have, whether they be sparse or tall grasses or ecosystems like that. As you can see in the top left, insects respond not only to vegetation structure, but also community composition of vegetation. So there's many insects, obviously, that are pollinators, and that feeds back into an essential service that insects provide on native grasslands. But as well, insects form a really essential component of the diet of grassland breeding birds. And I'm not going to get too much into that. Kevin will talk about that after me, but there are obviously really strong connections between insect abundance and bird diversity and abundance itself, and particularly in how birds, grassland breeding birds, are able to provide sustenance for their nestlings. All these things, we really don't know how these things connect intimately together, and so the whole idea of the project, as Diego pointed out, was to try to connect them all. So we collected insects in 3 different ways, really all for different purposes. So we did sweet net samples. You look at the top right sample, you can see in the top right picture, rather, you can see a picture of our sweet net, and see some examples of people sweet netting. Those are to collect insects that are higher up in the grass canopy, and they get at animals of insects that are really the focus of birds for as food. We also do a thing called tan trapping. You can see that little yellow disc. It's just a plastic tray that sits in the grass, and the yellow color will attract pollinators, and that obviously is a way to sample insects in grasslands that are going to be pollinating the various forbs that are growing in native grasslands. In our case, we use soapy water to trap the insects, and we put out an array of these plots, as you saw perhaps in the schematic that Diego put of our our radius plot sites. There's a line of these put out, a 15-meter line from the center of the radius plot to collect pollinating insects. And then on the bottom left-hand corner, there's an image of a tent there, and that's called a malaise trap. And it is collecting insects that are attracted essentially to light, and it is in all kinds of different attractants, to be honest. And we use those samples to do what's called metabarcoding, and I'll get a little bit into that later. Once we collect the samples in the field, we preserve them in a number of different ways. We either dry them or preserve them in alcohol, and then they're shipped off to a lab based in Ottawa at the National Collection in Ottawa, where the samples are identified, again, in 3 different ways. So some of them are morphologically identified. So we have an entomologist, Mrs. Benham, who's a huge part of our program here, and one of his lab technicians going through sorting out the samples. And those, so we can identify some of the taxa when we have expertise to do so using morphology. Otherwise, we use 2 other techniques that are genetic-based techniques. So one is polymerase chain reaction, or PCR, where we can identify individuals, usually down to family, but to some extent, if we have the right primers, we can get right down to genus or species. And that more or less confirms morphological identification. And then we've also been involved with a program at the University of Guelph to do metabarcoding. Metabarcoding is commonly known as DNA fingerprinting. You're basically taking, you're not actually analyzing the samples themselves, but you're using a reagent in which the samples have been soaked, and you put them through a DNA fingerprinting process, and you get a list of how many different insect and taxa occur in that sample. You end up with thousands of identifications. And so those are the 3 methods that we've used to determine the diversity and abundance of insects on our plots. We also do things like we collect dry weights and things like that from our samples as well. So I want to show you a little bit of the data that we've collected over the last, Diego's camera suddenly came on. Hey, Diego. Yeah, okay. So here's a little bit of some of the information that we've collected. So we're really interested in linking, obviously, our information about insects to some ecological variables, some environmental variables. The first one that we get is insect mass. It's a pretty easy measure to take. The identification takes a long time. In fact, we haven't even completed our 2022 identifications yet. We have the metabarcoding. We have some of the morphological analysis. We haven't completed the PCR analysis yet. It takes an enormous amount of time and effort to get that. So the real low-hanging fruit for us is simply how much mass is there when we weigh our sample. And you can see that we get a really nice signal with vegetation type. Insect mass tends to increase in areas that will tend to have thinner vegetation cover. And that's because if you think about what an insect is all about, they need heat and they like it dry in order to reproduce. They like those kind of habitats where you've got lots of dark surfaces, not a lot of shade, not a lot of water, and it allows them to go through their life stages much more quickly, and you end up with these much more abundant insect samples. And so you can see that in the bar graph in the top left, that thin soils where you're typically getting very little vegetation, that's where you're getting your highest insect mass. And then you can see in the plot on the bottom right, the more bare ground you have, the greater the insect mass. And this correlates well with what we understand about insect life histories. Now, we're going to get to this a little bit later. Obviously, the data for the scientists that are out there, you'll notice that we're really biased by a few sites that have very high amounts of bare ground on thin soils. Our loamy sites, the signal is less clear. So we're going to be moving forward to collect more samples in that area to try to confirm this relatively preliminary finding. We're also obviously very interested in pollination. And Benham is an expert in syrphids, which are also known as flower flies or hoverflies, you may know them as. They're really, they look like a bee. They're a bee mimic, but they're not a bee at all. They're a fly, so they don't have any stinging apparatus, but they're really super important pollinators. And of course, pollinators will respond to flowers. Native grasslands have an abundance of native flowers, and so we would expect to see some type of relationship between the abundance of forbs and the abundance and diversity of syrphids, of pollinators. And that's what we find. The relationship here isn't super strong, and we do get a little bit of a signal with different soil types that will link to the vegetation communities that are there. But what we're seeing is that positive relationship that we would expect to find between a pollinator and the abundance of grasses in that prairie, which we thought was very interesting. And here I'm comparing surface richness, which is simply, essentially it's a proxy for how many different species or how many different families in this case, or sorry, genera we're finding in plots. And so it's a count of the individual types of flower flies that we're finding in our plots relative to forb abundance. I want to talk a little bit about the meta-barcoding data. So we just got the meta-barcoding data back from our 2022 samples. It takes a long time. COVID didn't help us getting this through very well, very quickly. But you can see how much information you end up with. So we got 705 or almost 706,000 specimens were collected by the malaise traps in 38 different orders and 351 families. And there's a breakdown of the top 5 orders here. There's obviously many more. There's another 33 orders involved, but dominantly we're finding flies in our samples attracted to light, but we're also getting bees and wasps, lots of butterflies, some true bugs and beetles in our samples. So we get a huge amount of data, a lot of information, kind of overwhelming. And we're just starting to do some analysis, and I wanted to show you some preliminary plots. This is the 5 sites that where we collected data from 2022. And you can see that and if you start at the left-hand side and move right, you're actually moving further north. And we haven't done any data analysis in this yet. We just got the data a couple of weeks ago. This is the 5 sites that where we collected data from 2022. And you can see that and if you start at the left-hand side and move right, you're actually moving further north. And we haven't done any data analysis in this yet. We just got the data a couple of weeks ago. But the mean number of families is on the vertical axis, and then the site code, or as you move from south to the north, is on the horizontal axis. And you can see there's kind of a pattern where you have higher richness, more families as you move further north, which I find kind of interesting. Fair bit of variation in the data, variability in the data, and if you use that kind of old statistical rule of thumb where overlapping error bars will tell you something about whether there's statistical significance, it seems to suggest there's not a lot of statistical pattern here, but there's a signal. And so we're still beginning our work to explore where these patterns lie, which taxa would be most important in this relationship, but it's encouraging to see that we are getting some interesting results with respect to something that might be related to the environment. So a north-south gradient, for instance. Next steps in the insect research world. So we're still working on the PCR sequencing. We're trying out some new techniques with our colleagues at Agriculture Canada in Ottawa. And hopefully those will make future work in identification more efficient. We're doing some morphological identification. We still have some of that left to do. And then we feel that we have a pretty good sense. We have a lot of samples with respect to the biodiversity of insects on native grasslands. But we want to now move forward and really focus on that ecological interaction with birds and plants. And so we're going to be focusing our collections in the future on the 3 orders that tend to appear in bird diets most common. So those are the Orthoptera, the Lepidoptera, and the Coleoptera. So grasshoppers, caterpillars, and beetles. Grassland breeding birds tend to be feeding their nestlings these taxa, in large part because they're abundant, but in large part because they're they're large and they have a lot of nutrition. And so we've done some work with analyzing lipid contents. Kevin will talk about that in a couple of minutes, but that's going to be the focus as we move forward in the project. And I just wanted to acknowledge the many people who contributed to the insect collecting and analysis program. I'm not going to name everybody here. We've had really great field crews the last 2 years. We're going to continue our work this year. They have another great crew, and I will. And then at the end, we'll acknowledge all of the participating producers as well. But I just wanted to put a few names up there of the fine crews and lab technicians that we've had in this program. And I would hand it over to Kevin.
Okay, perfect. So thanks, Sean, and thank you everyone for coming to this webinar. So I'm going to be talking mainly about the bird angle of things, and showing some of our preliminary results. Again, we are interested in these drivers of density and diversity in grassland bird communities, specifically when it comes to habitat and prey. So what I'll be talking about today and showing preliminary results for are 4 different measures of habitat: grass cover, percent bare ground, vertical structure as measured by the Rebelco visual obstruction that Diego described, and then heterogeneity, so the standard deviation in those Rebelco measures. And then on the prey side of things, we'll be looking at insect biomass and insect quality as measured by their fatty acid content. First, to describe the bird data collection, it's a fairly simple protocol. So we do a point count at the center of each of these 200-meter radius plots. We truncate the data at 200 meters so that it matches the size of the plot. Each point count is 5 minutes in length. And we conduct 2 of these per season, and they both occur within the breeding bird period from late May to early July. And the results I'm going to show today are averaged across both of those visits. So the bird community metrics that I'm going to be focusing on generally rely on density estimation. So just to describe a little bit about how that's accomplished. So we use the QPAD framework that was developed by Peter Solomos et al. In this framework, you correct your raw counts into densities by accounting for detectability. And in this framework, detectability is broken down into 2 components: availability and perceptibility. And we're able to estimate these things from our data by collecting time removal and distance sampling data during our surveys. And this means that we sort our observations into time intervals and distance intervals in order to estimate those aspects of availability and perceptibility. And we also apply a pair correction to our densities using information from the Partners in Flight avian conservation database. The response covariates I'll be showing results for today are total bird density, species richness. I don't think either of those 2 require any more explanation. We're also using Shannon's diversity index, which is this measure of diversity that weights species evenness based on the intuitive idea that the more equal each species in the community is in abundance, the greater its diversity. And you can see how it's calculated in the formula there using the proportion of individuals that belong to each species. And then the final metric I'll be showing results for is the raw bird friendliness index, and I'll describe that a little bit more now. So we've been developing a bird friendliness index at Birds Canada over the last few field seasons. The idea is that it's a biodiversity metric that represents the value of the bird community on a farm or ranch. And the goal is to be able to use this to inform and evaluate land management on working landscapes and enable market and policy environments that incentivize good biodiversity outcomes in these habitats. The citation at the bottom of the slide there. So we based our bird friendliness index off of earlier work by the National Audubon Society in the United States, and you can check out that citation if you'd like more information there. And of course, the larger goal here is to try to halt the decline and reverse the decline of grassland birds, as well as shorebirds and aerial insectivores that use these grassland habitats. Now, kind of high-level view of the bird friendliness index. For today's purpose, the bird friendliness index is basically going to function as a weighted density index, where species are weighted by their conservation value. And we did this using 2 different criteria. One was the Partners in Flight Regional Combined Score, which they calculate for each species based on several different criteria involving population size and trend and so on. And then we also use our own multiplier based on species at risk status in Canada, which comes from Kasiwik. And this is a 1 to 4 scale where species that are not at risk or haven't yet been assessed by Kasiwik are given a 1, all the way up to a 4, which is for species that are listed as endangered. So kind of high-level results from the bird side of things. These are just the counts from our surveys that occurred at multi-taxa points in 2022 and 2023. You can see the species list there. Species that are marked with an asterisk are species that we use for bird friendliness index calculation. So you can see it's kind of reduced a reduced number of species. Now for the results I'm going to show today, we also remove species that aren't amenable to density estimation via point counts. So things like raptors, waterfowl, nocturnal species, which are not so detectable during the day, and so on. So just bear that in mind when we're interpreting the results, that the species list that we're using for the bird friendliness index is a smaller assortment of grassland birds than we're going to show for the other bird community metrics. So to dig into the results, so birds versus habitat. This is a kind of big, complicated figure. So we'll take it piece by piece. Basically, along the y-axis, you see our different bird community metrics, Shannon's diversity index, species richness, total density, and the raw bird friendliness index. And then across the x-axis, you see our measures of habitat, percent bare ground, percent grass, the visual obstruction representing that vertical structure, and then the standard deviation in that vertical structure as well. And the numbers that you see in the figure are just Pearson correlation coefficients. If you see any asterisks or periods after the number, that indicates a certain degree of statistical significance. The period indicates kind of weakly significant results between a p-value of 0.05 and 0.1. So just things to bear in mind as we go through these results. So breaking it down. So first looking at bare ground here. We generally find negative relationships between percent bare ground in an area and our measures of the bird community. This is a fairly expected result. Again, we see that the relationships are either not significant, not statistically significant, or marginally statistically significant. Bear in mind that this analysis involves just 47 data points. So we are a little bit short on sample size, and that's one of the things that we're hoping to resolve in the coming years, building up that sample size and being able to better resolve some of these relationships. But certainly we have some suggested relationships that match our expectation for what we should find. Next, looking at percent grass. So here we generally see positive relationships between percent grass cover and our bird community metrics. For vertical structure, we generally don't find any strong relationships here or suggested relationships here. This may be due to the fact, again, like the figure that Diego showed early on, where all the different grassland bird species kind of have their own niche within that grassland habitat as far as how tall the vegetation is. And so when we're looking at the community as a whole, we're looking at a very diverse assortment of species, and they all have their own habitat preferences, and we may not find the signal when we're looking at that larger community level. Now, when it comes to heterogeneity, so again, our idea here is that plots that have greater heterogeneity in vertical structure offer more different potential habitats to birds, and so you're maybe more likely to find more species, or maybe a single species will be able to satisfy all of its life history needs within that one habitat, again, because you have some variation, you have some diversity in habitat. And so here we don't tend to see very strong relationships for three of our bird community metrics, so the Shannon's diversity index, the species richness, and total bird density, but we do see a strong, significant relationship between that variation in vertical structure and the raw bird friendliness index, which potentially indicates that variation in vertical structure is important for species that are valued highly within the bird friendliness index, so species that have high conservation value. So just to summarize, so we generally found negative relationships between percent bare ground and our measures of the bird community. We found generally positive relationships between grass cover and the bird community. We didn't find very many strong relationships with those measures of vertical structure, except when it came to the standard deviation in those measures, so the variation in vertical structure within a plot, where we did find a strong positive relationship with the bird friendliness index, potentially suggested that those high value conservation species desire those kinds of habitats. Moving on to birds and insects. So a very similar figure here. So we have our bird community metrics along the y-axis again, and then we have our insect measures along the x-axis this time. The first three columns are different fatty acids that we measured within our insect samples. These are all essential fatty acids for all vertebrate life. They have particular function in terms of cell membranes, and they're known to be important specifically for birds as well. And basically in terms of how they relate to one another, so EPA and DHA are fatty acids that are like biological end products. And so the story here basically is that if organisms don't consume those directly, then they have to synthesize them from their precursor, ALA. Long story short, there's reason to expect that our various measures of the bird community would be positively related to these aspects of insect quality. So again, taking the figure one step at a time. First, looking at ALA. So here we generally find positive relationships between that insect lipid content and our measures of bird density and diversity. We find fairly similar results for EPA. Moving along to DHA. So one thing I'll mention as well is that for this analysis, the sample size is even more reduced because we only have the lipid data from 2022. Anyways, when it comes to DHA, so here we see that there is basically no suggestive relationships when it comes to measures of bird diversity, so the Shannon's diversity index and species richness, but we do see positive relationships between DHA content in the insects and measures of density. So total density, we see this positive but non-significant relationship. And then for the raw bird friendliness index, we actually see quite a strong positive relationship between DHA content in the insects and the raw bird friendliness index, which again may indicate that DHA content in insects is particularly important for bird species of high conservation value within these grassland habitats. Lastly, insect mass. So here again, we would probably expect to see some positive relationships between total insect mass on our plots and our various measures of the bird community. And we don't see that, which is a little bit surprising. We actually see these kind of weak negative relationships. And I think what's going on here is probably that we have some habitat confounds. So if we remember this figure that John showed earlier on, this positive relationship between insect biomass and the percent bare ground, we can see that insects seem to have some preference for these sparse bare ground kinds of habitats. And if we remember a couple slides back, in terms of how birds were related to percent bare ground, we found these negative relationships. So I think what's going on here is not that birds are avoiding areas with lots of insects. That doesn't really make much biological. It's just they don't like the same habitats that the insects seem to like. But overall, the result here is that there seem to be more suggestive relationships between vegetation and birds compared to particularly insect abundance and bird density and diversity. So just to summarize the insect results, we had these a negative relationship between insect mass and bird density and diversity, likely due to some kind of habitat confounder with the bare ground. And then we generally have positive relationships between insect lipid content and bird density and diversity, in particular that DHA content strongly related to the raw bird friendliness index score. And so I'm going to leave it there. I'd like to thank the Birds Canada field crews that collected the bird data here, and I'll toss it back to John for.
So yeah, I mean, as you can see, we're still in the analysis phase. We're still trying to figure out biological relationships, and as almost always the case in ecological communities, they're complicated. So it'd be super nice to be able to just say, oh yeah, we've got a habitat that has more insects, therefore there's going to be more birds, and this will be the places that have more grass. It's not quite that simple, and so we're still working on, as the title of the presentation suggested, we're still working on that sort of spatial scale component of it. And at the same time, the Multi-taxa Project, which was been going on for the last couple of years, is now been folded into the Living Labs Project, which is an Agriculture Canada sponsored program, of which there are many across the country and two in Saskatchewan. And we are using the information and the team that we have to study biodiversity on native grasslands to link that to the information that the Living Labs program is focused on, which is how active farming and ranching operations can have an impact on climate change and carbon sequestration. So we are beginning, well, we've already actually begun working quite extensively with Agriculture Canada and Cam Carlisle at the University of Alberta to link our biodiversity measures with soil carbon. You can see our soil, a punch truck with his crews sampling native prairie. And the one of the best practices that SODCAP is working with in the Living Labs Central Prairies program is to improve the diversity of crops that are put in tame pastures, the process called polycropping. And so instead of just having one or two crops, species in there to go up to 20 or so. And so we are doing some work, again, in collaboration with Agriculture Canada to look at, in my case, insects, but also sort of eventually bird biodiversity in polycrop sites as well. So that's kind of the future of the Multi-taxa program. And as Kevin pointed out, increasing our sample size is kind of key. So we find that our relationships can be weak, and we feel that by adding more samples to the data that we already have, we can improve on the outcomes of the statistical analysis and learn more about what these interactions are amongst vegetation, birds, and insects on the Canadian Great Plains. And my final slide is just acknowledgements. We really couldn't do this work without the producers participating, and this is a list of those producers and community pastures and Grasslands National Park that have participated in the program to date, and we really want to thank them, plus all of the people and organizations that have contributed to the Multi-taxa program. Thank you very much.
People and organizations that have contributed to the Multi-taxa program. Thank you very much. Back to you, Caitlin.
Well, first of all, thank all three of you thank you all so much for the awesome presentation. I learned a lot. It's really amazing the work that you're doing, and I'm really excited to hear more as your data sample increases. We do have some questions already from our attendees. So the first question I think I'll direct towards John.
Okay.
Ross is wondering if you have any idea how many insect families would be on annual cropland.
No, I mean, we might, someone might, I don't. We haven't been sampling on annual cropland.
Okay.
It'd be interesting. We would really like to be able to do that, to be honest, because I think, you know, the characteristics of insects that they like that kind of bare soil. At the outset of the season, I bet you there's be quite a few insects on annual crops. As the crop grows and that soil cools, then I suspect you will see a reduction. But I'm not an entomologist. I'm just sort of guessing from what I understand in the literature what might happen. But obviously, pesticide use is going to influence that, and that's something to consider. And we do work with, you know, we're working with Agriculture Canada to kind of tease some of those things apart. But it is a difficult question. But thanks for that question, Ross.
And Dwayne is wondering, did you measure rainfall in these areas? And that question came in during your presentation, John.
Oh, okay. No, we didn't directly, but we can get those data from, you know, we're scattered all over, as you saw from the maps, all over sort of southwestern Saskatchewan where the SODCAP area of interest is. And so we could get those from Environment Canada, but we didn't directly measure the rainfall, no.
Okay. And Michael's wondering, or he says, some of the correlations, for example, BFI versus insect mass, seem to be levered by just a few high mass observations on the right. If you look at the bulk of the insect mass observations, ignoring the outliers, do you see the same relationships?
Do you want to take that one, Kevin, or is that back to me?
Sure, for sure. Yeah, absolutely. And that is another kind of consequence of having kind of limited data set, that you do have those outlier values that are very influential. I can say that when you remove those outlier points, the relationships disappear. So you don't see those negative relationships anymore. You have kind of a null result essentially, which is not totally in line with what you'd expect. I think at the end of the day, we'd expect positive relationships between insect biomass and bird density and diversity. But at least it's not a negative result. So there is I think a bigger story to be told there. Hopefully as we get more data.
Interesting. And were forested areas within the grassland habitats considered separately, or was there no forested area?
There is. In some of the sites that we sampled, we did a little bit of work in the sand hills, and there's lots of aspen in those areas as well. We actually avoided those areas. Over the first 2 years of the project, we really wanted to focus on similar sites that had similar characteristics so that we could get that kind of baseline biodiversity data without confusing it with all kinds of other habitat types. So we don't have forested sites among our samples. They're all native mixed grass habitats.
Okay. And I just want to mention that Ross, who had the question earlier, Trevor Harriet says, email me that question. He'll pass it on to a friend at AAFC who might have an estimate on insect families and cropland. So
Okay.
Ross, if you're still on there, then reach out to Trevor. We have another question and I'm not sure who would be the best fit for it, but feel free to jump in. With increasing bare ground reducing birds but increasing insects, would this suggest that habitat is more important than food? It does look like it may be site characteristics, driving some of those patterns. Have you considered looking at the quadratic relationships between birds and insect abundance? And if you squint, it kind of looks like quadratic.
I'll start, and maybe Kevin will jump in. Yeah, we have thought about this a lot, actually, because some of the work that was done in the early 70s around bird feeding, John Weens's work, suggests that birds really aren't very selective when it comes to insect food. They'll just take what they can get, whichever is the most abundant. So that does suggest that maybe habitat is more important than food. I mean, ultimately, it's going to toggle, right? So if food becomes so limiting that birds are not able to successfully nest, then you could think that the birds might change their attitude towards what kind of food they're eating, or maybe forage further afield. But certainly, it seems to us too that the constraint of what kind of habitats to live and build a nest might be more important than the availability of food as the sort of the hierarchy of life history components plays out. I don't know, Kevin, do you want to add something more intelligible to that?
Yeah, I totally agree with that. Yeah, and I guess I just say that also, I mean, birds are mobile, so when it comes to their foraging, they're not necessarily, they don't have to be as tied to the area directly around their nest site either. And at least there's some evidence suggesting that at least some birds can be somewhat flexible in terms of their foraging areas, in terms of how they overlap with neighboring territories and so on. So yeah, I totally agree with John's sentiment there that that nesting habitat is probably the first limitation, and then they're a little bit more flexible when it comes to their diet.
Thank you for that answer. It's quite interesting. And someone else is wondering or mentions these data sets might do better in multidimensional analyses such as NMDS or PCA instead of the two-dimensional charts you showed. Has this been tried?
Yeah, that is a good point. We can do some multivariate analysis, but so far we didn't advance in this kind of analysis. But it's true, when you have many different variables, maybe we can put in one PCA or something to see different patterns. It's a good suggestion.
Okay. And Aaron says, I'm wondering if this study will be looking at carotenoids from insects as well. Any comments on that?
No. We're not really going down that road right now. Our focus is going to be on insects as bird food, which I know is a dissatisfactory answer for entomologists. I mean, insects are biodiversity unto themselves. They don't need to be classified as just a component of somebody else's diet in order to have importance. So I get that. But we don't have we've really feel that we've got a pretty strong sense of the biodiversity elements of insects on native grasslands per se. And so we're going to shift our analysis not to sort of more towards sort of those ecological interactions with the other components of biodiversity in grasslands.
Okay, perfect. And the next part of Aaron's question is, Similar to Douglas Tallamy's finding of 5,000 to 6,000 caterpillars per fledging, could this study determine a similar importance of grassland insect prey to bird reproduction?
In theory, it could. We would have to be estimating bird reproduction, though. We're not doing nest productivity as
Okay.
part of this. So the only measure of birds that we have are the counts, the point counts. So we don't have a
Okay.
nest productivity estimate.
Okay. And the last part from Aaron, arachnids are very important prey for birds, but apparently seem to be missing from your data or is minimal. Is that a seasonal factor or late summer perhaps, or?
Yeah, it could be a seasonal thing. Our review of the literature shows the same sort of thing, that arachnids do show up in bird diets. We do collect arachnids. They do show up in our samples. I didn't show the whole 38, all the different sort of families and orders that were that showed up in the analysis. They do occur. We don't think that they're a super important component of the bird diet during the nesting season, and that's the part that we're trying to capture. But they show up in our analysis, and we get all kinds of crazy things and certainly in the metabar coding, they show up for sure.
Okay, awesome. That's all the questions we have right now. So I'll give everyone a few minutes. If you have any more questions, feel free to type it in. And I just want to apologize. This was our first time trying multi-screen sharing. And I thought when I toggled, everyone would be able to see it. It didn't quite work out that way, but we've recorded the presentation. So if anyone missed anything, maybe Diego's slide at the beginning there, we'll have this available on YouTube later today. So you can rewatch it and catch he had some really good slides at the beginning there. So it'll be available there. Or if you know anyone that may have enjoyed this webinar, feel free to share it. And yeah, I'll just see if we've got any more questions that have come in. It doesn't look like it. So with that, I'll say thank you so much, Kevin, John, and Diego. That was a really fabulous presentation. And maybe when you've got some more results in a few years from now, we can touch base again and have you back and hear how your progress is going.
Sure. Next year we will have more data for sure from this season, so will be good. And if we can have a copy of these comments, Caitlin, will be good because are very useful for us.
Absolutely. Yeah, I can send that to you.
Thank you.
Perfect. Well, thanks so much, everyone. When you leave today's webinar, there's a quick 1-minute survey. So if you don't mind filling that out, that helps us report back to our sponsors so we can keep our speaker series going into the future. And yeah, thank you again for the fabulous presentation, Kevin, John, and Diego. And have a great rest of your day, everyone.
Thanks, Caitlin.
Yeah, thank you.
Bye.
Thank you.